Hi everyone, welcome back to the channel. Today I have another lesson plan, and this one is more targeted toward high school biology teachers or maybe undergraduate biology classrooms where you want to show the process of using some analysis techniques to look at variables in the context of biology. So what we'll end up doing in this example is looking at some data that are related in some way. We'll look to see if we can figure out a mathematical model to describe that relationship, and then we'll work on creating a nice visualization that is suitable for publication in something like a presentation. So the first thing is to talk about the data. Now, specifically, these data were obtained from Kaggle, which is a website that has lots of data resources. And I was able to track down the origin of this data set to a German publication from the mid-90s that translates to normal weight of the brain in adults in relation to age, sex, body height, and weight. So this was obtained through thousands of autopsies, and it's a little morbid, but the context here is a great example of two linear related variables, specifically the volume of the head and the mass of the brain. So to jump into this, first I'm loading a library called Tidyverse, which contains a lot of data manipulation tools as well as some visualization tools, and then another one that involves a statistical test that I'll use later on. So this is done in Quarto. That's the publication method you're looking at now, and the language I'm using is R. So the plan for today is to create a reproducible analysis with a well-designed visualization. So well-designed visualizations are great for reports, and the idea of reproducibility is huge in analysis. So if I were to do this and write down my code, I could give that to someone else and they should be able to run it exactly the same and get the same exact result. So for part one of this presentation, I'll be looking at the data and cleaning and formatting it as needed, and then doing a little exploratory analysis and adjustment. Part two is generating the model and examining the quality of the generated model. And then part three is creating that nice plot. There will be some plot generation in earlier steps, but they're more for on the fly, quick looks at the data just to see what's going on. So part one is to look at the data itself, bring it in, examine it. And so the first thing I'm doing is I'm loading up this CSV file that I've called brain mass, and it has a few variables in there. The first one is listed, and I got this description from the Kaggle page, but the first column is gender for male or female. Then the second column is age range, which is again two values, one for over 18 and two for under 18. And the head volume is a measurement given in cubic centimeters, and the brain weight is measured in grams. Now, brain weight is not quite accurate, so in my analysis I'm changing that to mass. So we have those variables. Here they are just as a quick preview in a tibble. So you can see that there are four columns with those names, and the benefit of a tibble is that it shows you the data type. So these are all listed as doubles. So they're all numeric values at this point. Now for me, I tend to like all lowercase names for columns that are all connected in some way, no spaces. And so what I do in this next step is just to unify all of the styles here. So all lowercase letters, I get rid of the spaces. And to do that, I just use an underscore instead of a space. And this is not completely necessary, but it avoids having to type the quotes around these names. So here we have the renamed file or renamed columns. And then I wanted to point out that because we have two of these columns that are essentially one or another value, they're factors. They're not really a numeric value. So the problem here, though, is that they're currently listed as doubles, which is a numeric value. So I need to change those to factors. So this next step is to use the as factor function just to cast these as a factor instead of a number. And so if we reprint that preview, we can see that the values have changed to factors. So at this point, we have all of the same columns, but they're renamed and the correct data types are present now. Now, for me, I don't need to look at all of these values. Currently, there are like 230 rows of data almost, but I don't need all of that. I'm going to actually restrict what I look at to a more specific slice of this data set. So here, I do a filter step, and this uses some of the capabilities in that tidyverse library. So I'm doing a filter and a drop in a step here to restrict the age range to 2, which is all individuals under 18 years. And then I'm getting rid of any rows that contain missing values. Now, I'm not really sure that there are any rows that have missing values, but just in case, that's my preferred method in this example to get rid of any potential issues. So now we're left with a data set that has 127 rows. And the next thing I want to talk about is assessing the quality of the data, because when you're looking at doing statistical analysis, there are assumptions about the underlying data. So if you have really poor quality data and you try to do a test and you get a number, you might not really be able to use that number. It's bad to trust that number if your data are not good coming in. So the first thing I want to do is think about the assumptions for a linear test, so a linear regression. So we need essentially normal data, that is that they follow the normal distribution. The variables need to be related to each other linearly. That is, if you were to plot them against each other, it doesn't form some curve or something else. And then the other assumption is that there is equal variance. And so we're going to look at all three of those. There are some others if you have a lot of variables, but in this case, it's a relatively simple test. So the main thing, again, we're looking for normal data, linear relationships, and equal variance. So the first thing we can do is look at the normality. And one way to do that is with this Shapiro test. So the test itself is relatively easy. We just need to provide to this test function the variable of interest. So here, the Shapiro test takes the head volume variable first, and we get this p-value of essentially 0.5. Then the next variable is the brain mass, and we get a p-value of 0.655. So both of these values exceed what is commonly used as a threshold, which is 0.05. And so in this particular case, the initial assumption is that the data are normally distributed. They're fine. They're okay. And so if this p-value appeared less than 0.05, we would have an issue, but we don't. So success on both of these. They both check out. We can assume everything's fine and these data are approximately normal. So the next thing I want to do is look at the correlation of these two variables. Now, this is not exactly a test of a linear relationship, but it's a helpful part of it. So we want to see if these two data variables are related. So the correlation test here is really easy. It's just COR for correlation, and we give it these two variables of head volume and brain mass. And then I'm just rounding the output here, so it's a little easier to read, but we end up with a value over 0.8. So that's a really good sign. That tells us that there seems to be a strong positive relationship. And in addition to that, I want to look at the data points themselves just to see if there is some weird curve relationship between them. You can still get high correlation values with nonlinear shapes in some cases. So this is just a good thing to do, just to take a peek at the data and see what they look like. So here we just use the base R graphics to do a really quick exam of this, and we see that the head volume and the brain mass appear to be linearly related to each other. There seems to be a little bit tighter grouping through the middle, and then maybe a little more spread at the end here. But overall, looks pretty good. Now, another thing to consider before we get into the analysis is outliers. There may be some values in here that are really outside the group in some way. And so when you do a linear regression, what you're essentially trying to do is find a line that passes through the data points to minimize the distance between those individual data points and that line. You're trying to make those residuals, as they're called, very small. And an outlier can skew that placement of the line, so having those in the data set is a problem. So we need to find out if there are any outliers and then do something about it. So the first thing I'm going to do is a box plot to look at each variable. And a box plot is a way to look at this distribution of values, and it shows these quartiles. And any outliers will show up as points outside of these quartiles. So first I'm looking at head volume, and I don't see any problems here. This is just a really quick snapshot with base R graphics, but any outliers would appear as dots above or below these quartile lines, but I don't see any problems. Looking at the brain mass, we can see there are 2 data points outside. So those are the outliers we need to look at. So for this, you can actually use that same box plot command to access the values of the outliers by adding this $sign and out at the end. So we'll use that same analysis to get them, and it reprints the plot when you do that, but it also provides these 2 values. In this case, 1588 and 1620. So these are the 2 numbers that are outside the group that we want to do something about. So in this case, I have a lot of data, so I'm just going to remove these. And the way that I do this is I find the index of that row that contains that value, and then I use that index and filter that out of the data set. So when I do this, I'm left with 125 rows. So at this point, just to recap, everything's been renamed, correctly set up as the right type of data. I got rid of the outliers, I checked the normality, and I checked the linear relationship, and everything's good at this point. So that's the end of part 1. And in part 2, we'll take a look at the linear model and construct it and take a look. So come back for part 2. If you like this video, make sure you like and subscribe, and I'll see you next time. Thanks.